And welcome back to Padashetti'S Pulmonology. And today's topic is Omicron. As WHO says, it's a new variant seen in South Africa. What I believe it could be just totally new virus, that is SARS-CoV-3. This is what I believe, not published anywhere. Okay. Without wasting much time, let us come into the discussion regarding this Omicron. We'll be discussing this topic under three headings: introduction, structure of Omicron, and mutation of Omicron compared to the original Wuhan strain, and regarding the vaccine immunity or vaccine efficacy. The scientific name is B11529, and the first official case, as you all know, was seen in South Africa on 24th November 2021. Until date, more than 80 countries have been affected so far, and slowly it is replacing the Delta variant. So as you can you can see how its transmissibility and infectivity are very high. So regarding coming to the structure, before going to the mutation, we'll study in brief regarding the structure. So it will be easy to study the mutation. There are totally 30,000 base pairs in SARS-CoV-2 virus. Okay, uh, base pairs, that is containing amino acids, are the basic pillars for the protein, and on these amino acids form four important proteins, and most important among them is spike protein. Why it is important? Because it is very highly antigenic, and most of the antibodies which are produced are against this spike protein, and it contains, and more than 10,000 of the more than 10% of the total base pairs. It contains around approximately 4,000 base pairs. So what are the two important parts? That is subunit 1 and subunit 2. This is a brief diagrammatic representation. This is S2, that is subunit 2. This is S1, that is subunit 1. And S1 has one more part, that is receptor binding domain. Receptor binding domain is a part of the SARS-CoV-2 virus, and the AC2 receptors, it is a part of the human cell membrane. Okay, this is receptor binding domain part of S1, and this is the ACE2 receptor. Okay, this is how the virus attaches to the ACE2 receptor. So what is the S1 helps in attaching the SARS-CoV-2 virus to the ACE2 receptor. What is the function of S2? S2 S2 helps in further entering or further fusion of the envelope protein with the cell membrane. Okay, cell membrane, human cell membrane. Okay, it further helps in fusion. Okay, S2 function is fusion of the SARS-CoV-2 19. S1 helps in attaching. S2 helps in fusion and further entering or penetration into the cell. And one more important point which you need to study is furin cleavage site. Furin cleavage site, you see furin cleavage site which binds together S1 and S2. Okay, and the trans membrane receptor protein 2, which is present on the human cell membrane, helps in cleaving this site. Once this site is cleaved, S1 and S2 are separated. S2 helps in further entering of the cell into the further entering of the virus into the cell. Clear? S1, S2 and furin cleavage site. Furin cleavage site has to be cleaved so that S1 and S2 get separated, and S2 helps in further entering of the virus into the cell. Why it is important? Coming to the mutation, Omicron has more than 60 mutations coming to the original Wuhan strain, and out of 60, more than 32 are present in spike protein, that is more than 50%. And out of 32, RBD, that is the receptor binding domain, has 15 mutations. This has 15 mutations, and additional 3 are present in this furin cleavage site, so that it gets cleaved early and infectivity rate increases. Okay, this is a mutation, and this is the total number of mutations which are produced, which are seen in Omicron variant. How do we read this? Okay, first you can concentrate on this one. It is A67B. A67B is arginine amino acid is replaced by valine in 67th position. Okay, that is what it means. Okay, that is how you read the total mutations. Okay, this is a Omicron total mutations. Now, after studying the mutation, we need to study regarding one more important topic, that is vaccine efficacy. It is very early to say anything whether the vaccines and the previous antibodies which are produced from the previous infection, whether they're efficient or not. But there is an important study which has come from Oxford University. What they have done is they have taken the serum from the patient who are previously vaccinated, not infected, previously vaccinated, and they have mixed it with the Omicron variant. What they have seen is there are reduced level of neutralizing antibodies which are produced against the Omicron. Okay, what they are seeing is reduced level of neutralizing antibodies which are produced against Omicron. This is a matter of concern. So this is what I believe. It is just a totally new virus rather than just a variant. This is what I believe, but there are many but we have to wait for a long time. That means we have to wait further to get updated regarding whether the vaccines and the post infection antibodies, whether they are efficient or no. Okay, this is regarding Omicron. If you have any comments, please let me know. This is very early to say anything. These are just till date. Okay, thank you all for patience listening.